So it's a great pleasure to have Dr. Chin with us today. Dr. Chin is a New Yorker, even though she did her earlier studies at Brown University, where she got a magna cum laude in her biology studies, where she was majoring in neurosciences. That brought her back to New York, where she did her medical studies at Albert Einstein College of Medicine, and stayed in New York to go on to pursue a residency training in dermatology with a great deal of interest in laboratory studies as well at Columbia University. She became the chief president at Columbia at the time. And Lynda started to do some very important translational studies. From New York, she moved to Harvard, where she became a member not only of the Cancer Center there and of the Melanoma program, but also at the Broad Institute, where a lot of hot data was coming up for deep sequencing and next generation technology coming up really quickly. And she pioneered some of these earlier studies with computational biology, became very involved with the Human Cancer Genome Atlas project that in part she guided mainly in the area of melanoma and melanocytic lesions. With all of that, Lynda moved to MD Anderson, where she became very much involved in the Institute for Applied Cancer Sciences. And in 2012, because of all of her work, both clinical, basic, and translational, Lynda became a member of the Institute of Medicine of the academies in the United States. Not happy just with that and all of the achievements, and as new developments were happening in her lab, she took more recently a very challenging position at the University of Texas, where she is the Vice Chancellor for an entire new area that is going to start transforming healthcare. Lynda has always been at the cutting edge. It's one of these clinicians that understands basic research, and putting it together has produced an incredible amount, not only of great information for helping us understanding disease, but also for better classifying and for the treatment of cancer. So Lynda, it's a great pleasure to have you here. And we want to give you just a little token. Dr. Klemperer was one of our esteemed colleagues and one of the great chairs of pathology in this institution, and this lecture is on his behalf. So we want to give you, just a token of recognition for being here today. Thank you.
Thank you.
Let's put it in here.
Carlos and I had met many, many years ago, and I want to add to Dr. Sin's talk earlier. Our first collaboration was in analysis of a mouse model, which was what turned out to prove that P16 is a melanoma suppressor, and Carlos was our pathologist. And as you know, it's not easy to diagnose mouse melanoma, especially back then. There was, 20 years ago? So we have collaborated extensively up to a few years ago with the move. And I'm going to, so we miss you, and I miss working in the clinical area, although I've been really in research for the last 18. But I'm going to take, I have slides, and I was changing them as I was hearing the talk, and I stopped changing them. I'm just going to skip over things that some of you have talked about. I wanted to give you a background on the genomic landscape of melanoma. I want to talk about some of our unpublished work in the area of epigenomics, because that really is another area in the genome that we need to begin to understand much more. But then I'm going to come back and touch on some of the newer work we're doing. As Carlos mentioned, I've become interested in how we can bridge the gap between between research and clinical practice, having experienced both ends in private practice. I used to have a private practice downtown in Manhattan, and academic research and clinical research. I thought that there's a big gap, and we heard about some of the challenge there from a therapeutic development perspective, but there's also a huge gaping hole in the area of biospecimen and human specimen analysis, and the value of that can bring to our research and translation. So I want to speak to that a little bit and end with what I won't talk about, but hopefully is provocative thought for you to take home with. So with that, I'm going to start, and clearly you have heard from several speakers already today that we believe today, I don't think I need to convince you that fundamentally cancer is a disease of genome, and if we could just understand all the things that have gone wrong in the cancer genome, then we could certainly do a better job in treating the patient, and there has been tremendous proof of principle of that in the last decade. But I think detection, and we have talked about and heard about the diagnosis and how molecular events can really help, and in the long run, the holy grail is really preventions. So I think there's huge incentive to get a better understanding of the genome, and that really was the fundamental driver of the Cancer Genome Atlas project, which you have heard alluded to. To me, that gave us what I describe as the genomic medicine paradigm. What we're learning is it's no longer enough to have a histopathological organ-based diagnosis. We need to understand the underlying genetics that's driving that particular subtype. But it is just as important that we have to have drugs that's developed for that subtype. And then finally, we have to have a way of identifying that responsive or excluding the non-responsive population. And it's only when you have all three of these come together when you start to have the clinical success, such as the BRAF inhibitor or the EGFR mutations in lung cancer. So you already heard, and I'm going to go through this very quickly, TCGA has taken on the study of melanoma as one of its 25 projects. And we have recently published the study after 5 years of work from over 300 investigators on multiple continents. And it has been difficult in many ways because the collection of sample, and this really summarized the tissue that we're able to collect, and majority of them are metastatic sample, regional, lymph node, and a few distant metastases, and then around 67 primary, and they're not matched. There are only 2 cases, so we have matched primary and metastases, so that's important to keep in mind. The analysis we've done was multidimensional, that's why I call them. In other words, we didn't just look at mutation status, we didn't just look at copy number alteration. We looked at expression, promoter DNA methylations, as well as targeted proteomic study, and each of the sample have rather limited, but some clinical annotations. And what we did was generate data on the entire cohort and analyze them. Now, what does that mean? Before I get there, though, I do want to give you a very high-level description of what the next-generation sequencing technology is, because that technology is what enables us to do the amount and volume of sequencing that we've been talking about. So for those of you who know, forgive me, but this is really the level of understanding you need to at least have with you if you want to understand sequencing. What next-gen sequencing, or really massively parallel sequencing, does is it fragments the DNA into relatively small pieces, and we isolate the ones that are about 400 base pairs. And what we did is the machine would sequence the 2N. At the very beginning, it was only able to sequence accurately about 21 base pairs of the 2Ns, and then it got back better. And most of the sequencing you will see in the publications these days, we read about 50 to 70 base pairs at either end of that 400 base pair fragment. And then what you do, you map it back to the reference, and that reference typically is a germline genome of that same individual. And if you now line them up and see a mismatch, and not just one fragment, but multiple fragments, because you have multiple allele of DNA that you have fragmented in sequence, statistically you begin to be able to call with confidence a certain mutation. And this is what we meant when we say depth of sequencing. So if you sequence very superficially, you don't have confidence, because given the 3 billion bases, there's that natural polymerase error. You're not going to be able to tell whether that mismatch is real or not. But if you sequence very deep, and that's when you hear about that, that's what it meant. You consistently see that mismatch. Now you know that's a mutation. You can also see missing chunks of DNA, a few base pair, a big chunk, like here, or you can see amplification when you see many more statistically. The most transformative well, one of the transformative capability next-gen sequencing offers is the detection of translocation. And what that meant is that you knew that piece of fragment, the fragment DNA you sequenced is about 400 base pair. But when the two ends map back into some region that's not 400 base pair contiguous sequences or on two different chromosomes, now you have a high throughput way of detecting translocation, either intrachromosomal or interchromosomal. And then finally, because you can read through most of the DNA bases, you can identify non-human sequences. And we all appreciate the role of virus in cancer. And certainly, I think this technology is going to allow us to identify many additional infectious etiology of cancers. Now, once you have all of the mutation, whether you sequence and analyze it in such a way to call mutation or call amplification and deletions, and that really gives you individual reading for each of the sample, and you compare what's in the tumor to what's in the germline, which is what you inherited from your parents, and that difference is the mutation that you're identifying. But we all know that many of these changes are based on noise and not pathological, so this is where the cohort analysis come in. When you begin to look at 100, 500, and 10,000 patients, you identify the event that are consistently changing statistically above the baseline, above the noise level, so that you know that event's likely, at least statistically likely to be biologically relevant. And that's how we get these data together. So with that paradigm, let's look at the data from the mutation analysis. And you've already seen this plot a few times. As you can see, when we look at all the mutations that are consistently, and we call significantly mutated, that are identified in this sample, we can group them. And you can see the biggest group is the BRAF hotspot mutation group. These are the V600 mutation, the NRAS, and you heard about the NF1. And all three of them do activate the MAP kinase pathway primarily. The triple wild type you already heard, and you can see that there are few mutations, at least statistically significant mutation, but we know even though with a cohort of 300 and some sample, that is a very small cohort considering the high basal mutation rate in melanoma due to the UV mutagenesis, which is being depicted here, down here. What this is telling you is a signature that reflects UV mutagenesis, particularly the C2T alterations. So given that, we know we don't have the power to detect the more rare events. That's when we do the comparison with a cosmic database where there's a curation of all the reported mutation out there in all the tumor, and that allow you to do a meta-analysis with a much larger cohort and identify rarer event, and that's where we can see the GNAQ and GNA11 mutation, for example. Now, I think the triple wild type really is, I hate to say it, the garbage bin. We couldn't define what they are, so we put everything else in there. Because they represent these various mutations that they clearly have different biologic function in terms of pathway activation. They also represent the subtype that have relatively high frequency of high amplitude alteration. And actually, Boris Bastian was the first one to describe that among acral melanoma that has high focal amplification of oncogenes, and we do see that, and they tend to be in the triple wild type subtype, which goes well with the fact that many of these have much lower UV signature mutations. Now, molecular, I think this is actually an important point. The genetic defined subtype that clearly reflect the pathogenesis. These are major driver during the transformation process, but they don't necessarily correlate with prognosis. And those are two different things. And what we can show is these subtypes don't necessarily have a better or worse prognosis, although the NF1 subtype is more prevalent, slightly more prevalent in the older populations, but really it's not a prognostic subtype. Instead, if we look at their gene expression, the transcriptome, and by the way, we look at other dimension of the genome, we didn't find any correlation, but in the transcriptome, you can identify 3 subtypes, and one of them clearly has a better prognosis in these patients, and these are pre-treatment samples. So, we have confounding factor in terms of how they are treated. The majority of these samples were all enough sample that they were predated the targeted therapies. And that in green is the subtype that's enriched for immune signature. And that can be replicated pathologically by the pathology team that went back to every sample and looked actually at the section and score lymphocyte infiltrate. And there's a very strong correlation between that gene expression signature reflecting immune presence, immune gene activations, as well as the number and amount of lymphocyte infiltrates. And therefore suggesting that sample, that tissue or tumors that have higher immune activations tend to have a better prognosis. Now it's important, careful in that wording is that I did not say that they are more likely to respond to immune checkpoint therapy, because we don't know that. You can perhaps hypothesize and then do a study to validate or invalidate that, but the correlation simply says that samples that have a higher immune activation status by these measures tend to have better prognosis. Now, this is one diagram that actually was quite useful to summarize the study we did in TCGA. As I said, we collected these samples. We were able to, based on these analysis, both pathology, molecular, and clinical annotations, identify these 4 major subtypes that we already talked about. One of the really interesting observations was clearly the immune signature that is correlated with prognosis, and whether it correlates with responsiveness to immune signature is going to be interesting, and I'm sure the answer to that question will come out. But the prognosis, as I said, is different from the genetic being able to inform you in terms of the choices of target therapy, such as MAP kinase or BRAF-specific inhibitor in the BRAF subtype, and so on. So with that, we clearly have now one of the most comprehensive collections of genomic data in melanoma. And by the way, the uveal melanoma project is progressing very well. I don't know whether anyone here is participating in the project. The analysis working group has started their analysis for the manuscript. So you can expect similarly rich information coming out for uveal melanoma. But we also know that epigenetics is important. Certainly over the last 5 years, there's a greater appreciation or acknowledgement that epigenetic modulator, the epigenome, isn't just a passive scaffold. It's actually an active participant in regulating genes. And here is a summary, several years outdated, that shows you various epigenetic regulators that are somatically mutated in a significant and biologically relevant fashion, suggesting altering the epigenome is a pathogenic path to development of cancer. So what we did was to begin to try to complement the genetic characterization of cancer with epigenomic. But the epigenome is a little bit more challenging than the genome because we don't have anything to compare to. Unlike the germline, which we can consider normal, clearly there are inherited susceptibility. That aside, we can compare a somatic genome to the germline genome and identify cancer or candidate cancer associated changes. We have not a way of doing that with the epigenome. You can't just isolate melanocytes from the skin, put them in culture. The minute you do that, you already change the epigenome. So what is normal? So so we had to come about it from a different way. So what we decided to do is we decided to do a longitudinal comparison to look at change. If we can define the epigenome at a certain state, that is a step before or after another state. We can compare these states and identify changes that are associated with the alteration. So for example, we can look at normal skin versus tumor versus metastatic lesions and identify, and those are very well-defined biological state. Then we can define the chromatin states. That's different between them. And that's what we decided to do, to get around the fact that we really don't have a good way to identify cancer-associated epigenomic alteration. The system we utilize is a human melanocyte that's been immortalized by TERT and inactivation of P53 and RB pathway, which is a well-known paradigm to transform human melanocyte, at least to prime them for transformation. These cells, if we just, then on top of that, we add in the signature BRAF mutations. They're not tumorigenic in the mouse. But if we then also knock down P10, and we know those 2 mutations cooperate in human melanoma, not surprisingly, now those melanocytes become tumorigenic in the mouse. And then if we activate the MET signaling pathway, they spontaneously metastasize in the mouse. So now we have an isogenic cell system that has 3 defined biological states: non-tumorigenic, tumorigenic, and metastatic. And this is just the in vivo study proving that. And we harvest tumor there, and then we can characterize them. And I'll tell you about our data in characterizing the epigenome in these 2 biological states. What we did was that we adopted a technology that allowed us to do high-throughput ChIP-seq all 36 chromatin marks simultaneously in the same sample, so that we're not just looking at any one single mark, but we're looking at least what we know to be a more complete set of the chromatin modifications, and did massively parallel sequencing to analyze these. So a single study here basically gave us about 3.6 billion reads, which is more than the entire epigenome roadmap that was published about 3 years ago. There's a whole lot of data. I'm just give you 2 little tidbits to give you a sense of what we can do with this kind of data. With all 36 marked, obviously we can consider them individually as if they have no relationship to each other, and just compare how the mark behave at each position of the genome in the non-tumorigenic and tumorigenic state. And this is simply what it shows. It shows in the histogram that there's a change in red, which are the acylation mark, and then a couple of the H3K4 methylation mark that tend to be different. Now, we also have more sophisticated analysis where we consider the fact that these marks don't exist in isolation. They're actually in combination, but I'm not going to go into that. But in the most simple level, we simply compare all 36 marks to each other across a 2 sets of cell system and identify acylation mark being one of the common changes. And then we look at the genes where we see the promoter acylation being different between non-tumorigenic and tumorigenic cell, because these, in the literature, these alterations in the promoter is known to regulate gene expression. And you can see that, that there's acylation changes, and then we can look at and basically map of all the genes whose promoter have the alteration in the acylation mark at the promoter. You can see that some of the genes had deacylation but had upregulation of expression, and other had the opposite, downregulation of expression, and some of them don't change. So now that gives us different subsets of candidates. And what I try to tell you, if you didn't count the number of dots, is that there are way too many for us to do anything about it. So this is where you have to get a little smarter. And that's when we say, okay, we got to do something to filter this down, because I don't know what else to do with that. We decided to integrate a functional genomic screen in vivo in the same system. We took advantage of the fact that we know the BRAF transform prime, BRAF prime human melanocytes are not tumorigenic in vivo, but when they have P10 knockdown, they become. So they are prime and ready to be. Then we can introduce a library of epigenomic regulators and ask which of those regulators can cooperate and take their cells to the next level so that it actually can be tumorigenic in vivo. And we did the study both ways. We overexpressed the regulator, and we knocked out with hairpins. And this is a result from our loss of function study, where 5 hairpins that's validated for each of the regulator, there are 95 of them, were generated in a library, introduced in vivo, and allow the tumor to form. So your background is, and this is called gain of function screen, the background is you don't get cancer. There's no tumor. When you add something to it, tumor grows, so you harvest the tumor. So it's a gain of function. Now what you can look in the tumor is what has been enriched. What did you take from that library that allow you to become a tumor, and that becomes your candidate. And that's exactly how we identify. And what we did was we identify a series of hits that can cooperate in that context to generate tumor within the same time frame. And then we have to then go back and do secondary validation. So here are all the validated hits. And interestingly, as you can see, they match us up with the epigenomic profile alteration we identified, because 2 of them happen to be acetyltransferases that regulate the acylation marks. So now we can begin to narrow down, because we now know which 2 enzymes that regulate that mark are also oncogenic, or actually tumorigenic, because this is the loss of knockdown screen. It's a loss of function screen for gain of function. It's a little confusing. And now give us a better handle now to narrow all of those candidates and begin to understand what the likely changes are. Now, I can tell you that, for example, we now know that one of these is validated, TAF1, for example, and it is an enzyme, is druggable. And you can begin to imagine how that you can quickly get to a point where you can take massive genomic and epigenomic profile data, take advantage of defined biological state in the isogenic cell system, and combine that with in vivo functional screen in one experimental system, identify very manageable number of candidates that are therapeutic targets. Now, who's going to ask me the question? I'm going to skip over this one. What's that have to do with humans? I've just told you I did a cell-based study in culture. I did an in vivo screen in a mouse, and I also did a lot of profiling study. But still, it really hasn't proven the human relevance of it. Because in my view, every time you do a candidate discovery study, validation must include validation of human relevance. And that's really important. So how do we go about doing that in this case? So we decided to take advantage of the fact that the non-tumorigenic state and the tumorigenic state correlate with the benign nevi and the melanomas in human samples. So what we did was we collected a handful, not enough of them, I would say, but at least we started with a handful of benign nevi. I think there's a number here. 4 benign nevi and 9 melanoma tumors. And then we also devised a nanostring-based chip assay. Instead of sequencing a whole genome of all those 13 samples, because it's a lot of sequencing, if we were to do the same experiment we did in the discovery phase, we devised an assay for validation, and the assay is done on the nanostring machine. You must have used those, and they're pretty easy to use. It's 96-well assay. We statistically select 6 antibody, 4 sets of region across the genome that have variable changes, the same way you would select signature, and look at a bunch of acceleration marks as the primary mark that we're focusing on. And what I'm showing you here is that once we generate that profile of 96 data points, we cluster them, and what you're seeing is that under this branch, red and green go together, and red happen to be non-tumorigenic cells, and green happen to be the benign nevi. In contrast, the orange is the tumorigenic state, the tumorigenic cells, and the blues are the melanoma, and they're closer together. Now, you can see that better when we do principal component analysis, and you can see how these cluster together in the 3-dimensional distance much closer, the green and the red. Now, interestingly, I can't say the same about the melanoma sample. They are definitely not clustering. I can say they're farther away from the nevi, but they are not clustering together. So there are 2 explanations. One, we did not pick the correct region to look at, or the correct marks to look at, because we're selecting a subset of data we have to ask that question. Or more likely, we're seeing the same heterogeneity we expect and we have observed on the genome level among tumor that you don't see in benign neoplasm on the same epigenomic level. So how are we going to figure that out? This is what we're doing now. We collected about 25 samples that have been through TCGA analysis melanoma. These are real human samples now, full tumor sample. Technically, it has been challenging, but we are optimizing, and we finally begin to actually be able to do the same high-throughput ChIP-seq in whole human tumor sample with down to 3 milligrams of tissue. Still significant, but much less, because up to now, we haven't been able to do ChIP-seq on anything other than cells, because we couldn't get enough input material. So the optimization, and I think that technology will continue to improve, and we will soon have a complete epigenomic profile on about 25 sample or 40 sample, I can't remember, that have full genomic characterization. And that will give us our first data to begin to understand what the genome, the relationship between the genome and the epigenomes are. And so we're excited about that. But I would tell you, however, we're absolutely facing a major challenge on the analytic side, because when it comes to this level of epigenomic data, it reminded me 15 years ago, there was no algorithm to analyze copy number alterations. And that's what we're facing now. We will have to deal with the analytic challenge of copy number epigenomic alterations. So I'm going to now switch, and the timer hasn't gone anywhere, so I won't know how I'm doing with time. Now, I'm going to switch gear a little bit and talk about phenotype. I think this is the waterfall plot that you're all very familiar with and very proud of. This is the BRAF, targeted BRAF inhibitor in BRAF mutant melanoma. But you can see that clearly, even among the responders, they don't respond similarly. And so I think the most common phenotype probably is the fact that they're variable in response and in outcome, even though based on everything we know, they supposedly have the same diagnosis. So if you take the BRAF mutant melanoma, there are a particular staging. What we see, if you look at all the response from the patient, they really feel like this graph rather than this graph. And by the way, this is sort of middle of the road. If you go through your textbook, the wiring of the RAS and RAF signaling pathway, there's the really simple one, and then there's one that's like a full page long. I took one in the middle. Well, let's just say none of it works that way, because they all work more like this way. Because in vivo, in the human, it's actually much more of a complicated, multidimensional, and dynamic network that you could never expect. Because if you look at this diagram, you may say you have BRAF mutants. Mutation in your tumor, you should act this way. You should behave this way, and I expect that. And we know that doesn't happen. And instead, it looked like this. Why is that? Well, that's because, let's look at the situation. This is a representation of the genomic data we get from a sample. It says you have a certain alteration, translocation, and mutation, and so on. And let's say that all the sample have BRAF mutation here. First of all, we don't know what else they have in the rest of the genome. We already know they have different things in the rest of the genome. On top of that, these tumors don't live by themselves. They function in the context of a microenvironment that have many other things going on it. And the many other things in the host microenvironment is very much dictated by your second genome, which is the germline. We often forget how important that is. We heard about the GWAS study telling us association, but unfortunately the world of GWAS and the world of somatic genomics, they're in two different worlds. And they can't be. They got to come together because certainly in real life, they don't function separately. And then I would say we can easily add the microbiome. There's enough data for us to say that's minimally your third genome to deal with, and probably many more. And then on top of that, your environmental exposure. That we all know influence, certainly if you look at sun exposure, is a great example that environmental exposure influences how your genome, your germline genome reacts, and that will influence how your somatic genome responds. So all of these go together and give you the phenotype. So if you ever expect your BRAF mutant melanoma patient to behave the same, then you shouldn't be, because you should never expect it to. So how are you going to begin to dissect and understand, get to the next level of detail of being able to parse out the association between phenotype and genotype? Right now, I say we are really good at, on very high level, segmenting our patient population. We're nowhere near, and I don't use that word for that reason, personalizing. I think we're lying to ourselves and to everybody else, because you just have to look at this plot. So what do we do? Well, the first step is we have to start bringing data together, because just like the GWAS and the somatic genomics communities, if those data don't come together, we're never going to be able to figure out that interaction between those two genomes. Similarly, look at the TCGA data. We spend millions of dollars generating multidimensional genomic characterization of these precious samples, and we have 10, 20 clinical fields that are being populated and annotated. And we all know that those are barely touching the surface of characterizing what that patient is, and we barely know what the outcome really is. Not to mention, you are, as much as I took time to build our disease working group and our clinician expert that came together and defined those clinical fields that need to be pull from medical record laboriously. Our expert only knows what our expert knows. The whole point here is that we want to know about things that they don't know. So they wouldn't know what else to annotate. So fundamentally, this is a flawed system. We will never get to the bottom of it because we're constrained by the box that we're staying in. The only thing we'll ever get is incremental knowledge. But what we need to get is outside the box. And the first thing we have to do is we need full clinical information on everybody, not the subset that you think is important or you think is important, but everything. Because the thing you don't know doesn't actually, someone was just talking about that. Just because you don't know doesn't mean it's not important. So we got to have all that data together, and it has to be real time. It can't be what I call sample of irrelevance in the freezer. We want actually real sample in real patient that's undergoing real treatment. So what we decided to do the last several years is think about how to build that infrastructure that allow us to integrate research and clinical care in such a way that it should never be in the past. I don't believe, and I'm a firm believer, that science is not medicine. Science cannot be in the critical path of medicine, but the two cannot be too far apart. So how do we build an infrastructure that they can parallel track? So this is our Apollo platform. And there's a piece that we wrote that is coming out in Trends in Cancer that really describe the philosophy and how we built this. But this is a graphic outline of what we try to do. The concept of approaching patient and generating the consent, or well, first of all, getting your consent approved by RIB, and then approaching them, collecting sample, banking them, tracking them, and generating data. That is, by the way, about 80% of the time and effort most translational researchers spend their time on, and none of it is science. So we really shouldn't be doing that. It's a waste of money and time for any one of us to be doing that. That needs to be centralized. Not to mention, from a quality perspective, that needs to be standardized. If we don't know where your sample comes from, how do I know that data can be combined, right? So we first started out with standardizing end-to-end process of consenting patients and having a dedicated team who really mapped out, do you know how you're tracking the sample? How are you labeling them? Are you sure that it is what you think it is when it shows up on the other side of the lab and turn into data? So we have to have confidence. It's no longer the 10 sample you're working with. We're trying to collect 10,000 samples. So we have to have that process in place. And then we allow care to happen, and that's what happened. You go to a lab, you do lab tests, and there are all sorts of stuff, and there will be treatment decision, and there will be outcome being measured. All of that is in your clinical care system, which is very much separate from your research database. We need to bring them together, and that's our big data interchange. We have to intentionally link with PHI all the research data and the clinical data. The whole concept of de-identified research is fundamentally a barrier. We can't do translational clinical research if all our samples are de-identified. So we have to create that. Now, that's not to say we can't put the access control and the process and governance on top of it to make sure the data is used properly. But we shouldn't make it de-identified. Give you an example. MD Anderson had over 1,000 samples contributed to TCGA. And the whole, there's one premises, especially early on in TCGA, going to the institution and convincing investigators to contribute their sample to TCGA. And one of the carrots is, but you will have the clinical data. You will get this free genomic data, and you'll be able to do a level analysis no one else would be able to, even though the rest of the data is public. Well, guess what? There was an attempt to try to re-identify that 1,000 sample in TCGA from MD Anderson, within MD Anderson, to the patient record. It is so well done that it's not possible. What's the point? So I think we need to build an infrastructure where it actually should be linked. So the intent of big data interchange is that there is full integration of the research and clinical data, but we can put all the control in place. And then now we can put analytic on there, and we can also bring in outside third party like TCGA public data. So we actually did run 2 pilot studies. What did I have? On this one, we started in leukemia. We get a IRB-approved protocol to do serial biopsy. You might say, okay, that's not hard in leukemia. That's standard practice. That's why we started from there. We show that we can get all the leukemia docs to agree that there's a common consent instead of everybody fighting for their patient for their only their own protocol that's going to analyze 5 molecules, and now you wasted the rest of the sample for everything else that someone else could have generated data and used for. We centralized that. All the sample were processed the same way in parallel. We only split them from our clinical path when we need them. Otherwise, we do one DNA isolation rather than 2, and so on. And then we integrate all the full longitudinal clinical data from these patients with their research data, which is whole exome sequencing. The second pilot we did after that, because everybody said, well, that's because it's leukemia. So we had to go to solid tumor. So we went to melanoma. We, again, got a serial longitudinal biopsy consent protocol through IRB with a lot of work, and started to approaching same way, multiple biopsy in patient. And in this case, in addition to genomic sequencing on these melanoma sample, we also now put them through immunotherapy profiling platform that Jim Allison started to characterize the immune profile of these samples, so that now we have 2 research data type linked to the full clinical information. So today, we now have, and I really do believe is the first, fully integrated research and clinical research database at MD Anderson, where we have over 165,000 patients, longitudinal full clinical data, linked to whatever research data we have on them. Here's one example. This is the immune profiling study we did in melanoma, particularly in patients that were put on CTLA-4 blockade initially, and some of those responded, and some of those, majority of those relapsed, that were then put on PD-1 blockade, and then some of those responded and some of them don't. So as we all expect, but we're able to identify the patient, approach them with our longitudinal consent biopsy protocol, and collect the sample, and generated genomic and immune profiling data on this. And this really has been tremendous work by the clinical ops team, but really spearheaded by Jennifer Wago, and some of you I'm sure know her, really a force nature who made this happen. So here are multiple time points, as I said, and I'm not going to, again, totally unpublished, very, very hot, in fact, new data from last week that we're hoping to be able to submit soon, but just a couple interesting observations, just because we have the ability to do the kind of data. And by the way, we consented accrue over 400 patients in 9 months, because we try to standardize the process, and we have over 300 and some sample collected, and I believe most of them, over 70% of them, have multiple samples. So obviously, the longer we keep this going, the more we're going to be able to collect. So the first thing I'm going to show you is, now we put all these tumor biopsy into a panel, and we did 2 immune profiling panels. One is standard IHC through the immune profiling platform, and this is the markers that were done by IHC, but there's also a larger panel that were done by flow. And we're looking at that in comparison. And then we have a nanostrength gene chip assay that really focuses on genes that are immune related. And then we have a larger genome-wide study on the sample that we have enough tissues left. So interestingly, what we found is the on-treatment biopsy is what predicts the response, not the pre-treatment. And perhaps that really intuitively shouldn't be surprising. There's a baseline level, but it's how that spaghetti ball of a network respond to the drug that tells you whether that is responding. So what we're seeing is on treatment, early on treatment, after the second or third cycle, CD8, CD4, and PD-L1 expression is, but not the pretreatment to CTLA-4 blockade. And same also observed in response to PD-1, and this is just the IHC images themselves showing that there's a clear difference in the on-treatment sample, but we don't see this in the pretreatment sample. Now, the interesting thing is that if you start comparing from patient that responded to one immune checkpoint therapy versus the other immune checkpoint therapy, we do see a difference, and this is on the level of gene expression. We see a difference in their signature, suggesting that the biology between CTLA-4 blockade and PD-1 blockade is different, which shouldn't be surprising. And because of that, we will see a difference. And that's important because that gives I think, the beginning of that scientific basis for combination. Even though we already know clinically combination gives us better and more durable response, great. But this is now the scientific evidence that will come out behind it, proving that there's rationalization why that should work, and that will hopefully going forward guide us in additional combination and rational combination, because I think we'll get to the same point, just like what we do today with targeted therapy. It isn't just combination of everything. There's a rational way of combining them, and we will see the same thing with immune checkpoint therapy. So I'm going to end now with the last thing that we've been doing, which is more food for thought for you to take with you. I remember the day, and I don't think I'm that old. I remember the day when, the concept of a tumor suppressor gene in a tumor type. I remember the day when BRAF was sequenced and identified as a mutation, and then the targeted therapy. It really happened very rapidly, and I would say, without offending anyone in this room, majority of our practicing oncologists were not trained in the era of targeted therapy, genomic-based medicine, or immune checkpoint therapy. And I don't know about you, I certainly would feel me prescribing a drug I've never used once before versus something that I was trained and done it for years, very different. So adoption of new therapy, particularly today when we're seeing new things coming out, not every 6 years, but every 6 months. How are we going to make that happen? Because the patient, honestly, 90% of them will not be seen at your institution nor at my own institution, even though I'm not there anymore. 90% of them will never be seen there. How are they going to benefit from all the great stuff that we talk about here today? So there's clearly a gap in adoption. And we all know with EGFR inhibitor in lung cancer, we know it's a 7-year, 12% adoption in the community setting, according to ASCO. So that is not that great for the patient. So now, you can come to a meeting like this and learn. We know the challenge to that because, that's time away. And then there's real, having heard about it versus having the support that allow you to actually use it. Totally two different things. So it would be great, you guys all, the residents, you're in the program, you can go down the hall and ask experts. But most of the practice oncologists can't unless you, happen to have a friend and know their phone numbers. They answer your phone. So the idea is that how can we perhaps make that expertise, that up-to-date knowledge be available in such a way that every practicing oncologist can access it so that we can accelerate adoptions. And so about 2.5 years ago, I began this collaboration with IBM Watson to develop a virtual expert system that I call the Oncology Expert Advisor. Now, it's trained much the way we think about training the fellow. The fellow come into your fellowship with their basic knowledge, and then they go on the ward following the attending around, and they sort of tell you what they do, and then the attending come in and say, no, you're wrong, watch me. And a year later, now you're the expert. So that's how we train our fellow. This is actually the way we disseminate knowledge today. It's kind of old-fashioned and sadly very low fidelity, and certainly not scalable. So what we did with the cognitive system that we built is that we fed them the basic knowledge, NCCN guideline, the textbook, the basic literature, and then we show them the actual cases, and then have them give the recommendation, and then we have an expert come in and say, that's wrong, that's right, and we just go through that process. The difference is that once we finish, these guys is 24/7. They actually can be available. So that's the idea, and you can have almost instant incorporation of the latest paradigm, latest evidence into that recommendation engines, so that it isn't about telling you what to do, but it's about, it simulates the concept that actually, this is another way of showing it. It simulates the experience of calling up your attending or the expert to ask. Instead of you knowing which chapter you have to go to look up. You say, hey, this is my patient, 56-year-old. This is what I know about him. This is the path. This is the lab. What would you do? So what you get back is information that's relevant to that profile of the patients, so that you are not looking through the Google search. You don't have time, or the textbooks. But you're looking at things that shows it, but is linked to the publication, whether it's NCCN guideline or the published literature that says this is appropriate. Now, it will be multiple options, because we only know so much about the patient, and there are real things that would never go into the EHR or that we ever train OEA to consider, at least not yet. So, but at least it presents to you the latest knowledge. And what I hope is that this allow our practicing physician to make decisions for their patient based on the latest knowledge base, rather than knowledge base they were trained with. And that's the gap that we're trying to bridge. And by the way, I have this as a demo on my iPad for anyone who wants to see it. And what we wanted to do really with that as a starting point is the concept of bringing the expertise to the patient, because we know 90% of this patient would never get the luxury, for one reason or the other, to come to an academic institution, come to a specialist to get the type of treatment that one can expect at a place like that. So we need to take the expertise out to those community practicing oncologists, so that those patients who are being treated there actually will be able to get that benefit. So finally, from there, as Carlos said, I moved on in the last 6 months, because I decided that that challenge I just talked about in terms of the gap in our delivery system, in our knowledge base, isn't specific and limited to cancer. It really is fundamentally a flaw in our current healthcare system. And so we have now started to take some of these technology, or I should say, building on the technology we have developed the last several years, started to take this to the other end of the care delivery spectrum, which is population health, and begin to develop the same system for that, so that together, when we look at the two ends of that spectrum, we can imagine a more person-centric care delivery system that's for health and health management, rather than simply is really optimized for acute care only, which is our current provider-centric model. This will impact you, assuming I'm somewhat successful. But me or someone else, this will happen. The customer's person-centric approach to life is happening in every aspect. Take retail. You don't have to go to the store anymore. The product can come to you. This will happen, and this will change your practice. And it is important for you to start thinking about how you can be on the leading edge of that change, rather than struggling to follow. I'll end, and here's a long list of participants in the TCH Melanoma project. And this is really, I don't think this is everyone, but it says everyone, so I would trust it. We have over 300 participants that took 5 years of work to do that study. Here's some acknowledgement of my old labs. Kunal Rai is now an assistant professor who really led the study in epigenomic that I tell you about in collaboration with Jason Ernst from UCLA. And I tell you about our immunotherapies study on the checkpoint, which is a pathology fellow, actually a Dermatopath fellow, and her projects in collaboration with Jen Wago. I mentioned about our work on the Apollo Big Data platform, and that's absolutely a multidisciplinary and cross-institution collaborations, particularly with IT and clinical op and legal and compliance, don't forget them. And then finally, the network democratization is a cross-industry collaboration with my partner being IBM, AT&T, and PricewaterhouseCoopers. I'll take questions now. Thank you.
I have a question. Do you think it's possible that the stated efficacy of immune therapies such as anti-PD-1, anti-PD-L1 antibodies may be confounded by the fact that those patients who are thought to respond or have had a greater degree of lymphocyte infiltration to begin with and may have had a more indolent course to start with?
You're talking about the untreated sample, or you're
Yes.
talking about the PD?
No, I'm talking about initially the disorder is heterogeneous, so say melanoma. some incipient neoplasms in the early stage have infiltration of lymphocytes, some don't. It's, I think, well established in many paradigms in organs, not just in cutaneous melanomas. The degree of the initial lymphocyte infiltration signifies better prognosis. So what I'm saying is, could that be in part what compounds the efficacy of the immune therapy?
Oh, okay. Well, I probably shouldn't try to answer the question because I'm not an immunologist. I think it's possible, and we don't have data yet to definitively address the question you're asking. The fact that we are seeing a difference. No, actually, I don't think I have data to address your question. So I agree, but we haven't. The only thing I would say is that we haven't actually proved that the initial baseline activation of the immune system that led to overall better prognosis is actually the same immune activity that is responsible for response to checkpoint therapy. I think I need an immunologist to answer
I'm not
that
just saying
question.
this to be devil's advocate, but I'm saying it because some proportion of cases that are deemed clinically responsive, it's known that their disease is not cleared by imaging. Rather, it sits there for a very long time,
Stable.
and nothing happens. Okay.
Yes.
On the patients who have the lymphocyte response, it's CD8 cells that are generated. And isn't that like the TILs that Patrick makes in the lab that fight the melanoma? So how can we, get those
Are they the
two?
same, you're asking?
I think they probably are.
I think that is the study that they are trying to address that question.
And secondly, I have a lot of patients who've had multiple melanomas, like 10, 12, and they've had multiple dysplastic nevi removed. It seems like that would be a good control population. The nevi in the people who have had multiple melanomas of different lengths. So when you first came, I offered to get those for you. I think now with the epigenetics, that would be an important group of people to study.
Yes, and I think so. But my bet is they're different because we actually have data where from the same patient, different lesions have different profiles. And clinically, you do see different response. So that's not surprising. But we have not done enough of study to be definitive about that. But your collection may be really helpful for that.
So we need to figure out how to get it.
Yes.
I don't know if I had... Hello? I don't know, perhaps I misunderstood you. You were talking about de-identifying data. Because the IRB, whenever you do a study here, they make such a big deal about it, and it's always couched as an ethical issue. Perhaps it's just the opposite. Perhaps by de-identifying, you're not helping the patients. I mean, that's what you're implying, isn't it? I mean, that's what I took.
So, okay, I think I do believe HIPAA is misinterpreted to a great extent, but I don't have any say over the law.
Because I can't tell you how many times we filled out an IRB, and they made such a big deal about that.
Right.
And you have to have a neutral observer, and you have to have somebody else code it, and all this other stuff. And it's really very difficult to do. And sometimes you actually may be doing a disservice to the patients.
Well, I believe we are.
Residents that we asked to do this. I'd never do it myself. I have somebody else do it. But still,
Yes.
okay, I agree with you completely. If
So
that's what
this,
you're implying.
I'd like to, and we have finally, we have our, so there are control that we try to put in place to address the IRB's concern, because we have to go through that process and a lot of education along the way. What we make the case is that the truth is today in your translational clinical research, people are using link information, except they're doing manually. They have their own little Excel file or whatever database they have that's their research data. They send their residents to go and look up the clinical information and link them together, and under IRB approval or not. But there's certainly an exploratory phase, preparatory to research that were happening before there's an IRB protocol because by definition that they ask you to provide the hypothesis, the number of cases, the statistic power, all the things that you know you have to put in your RIB protocol means you have to have an idea what you're going after. To get the idea, you have been doing preparatory to research study, which are linking research data with clinical data with our protocol. But the difference is under HIPAA, and we spent a lot of time with the lawyers on this, is that phase is allowed. It is allowed, in reality, under law, because you have to have that. But if you now want to go and prove a hypothesis, you have to have validation. So we built the big data platform as the full integrated data is only allowed for use for preparatory to research. So the minute you want to publish that data, it could be individual patient data, or the minute you want to do a validation study, which you will have to for any hypothesis, you need to go back to the old way. You have to have your hypothesis go through IRB and get approval. So what that means is that we have to build into the big data platform enough control to give confidence that the data is going to be used in such a way that by default is de-identified. So in other words, when I say they are identified, they're fully linked. That doesn't mean you have to show the PHI data to anyone who log on. That's simply a control you put into your system. And it's actually a lot easier to do than the way we're currently doing it. Because currently you're relying on the person. Remember that they shouldn't be showing the data to someone else. But you can, on a system level, with a line of code, not allow any PHI data to be shown. But behind it, every piece of research data is linked to every piece of clinical data. You're just not allowing PHI to be shown. When someone requests and needs a PHI data, whether for any reason, now you have to go through the IRB. So that's the allow activity on the big data. And under that, I think the other really important point of the big data policy the institution is embracing is that data, that preparatory to research opportunity should be available to every legitimate researcher, MD Anderson. It is not limited to the person who accrued the patient, who generated data, who consent, none of that. As long as you're a legitimate researcher, that data needs to be available, because that's the only way we can accelerate the translation. The proprietary nature, ownership nature, and frankly, we have no right to own them, but we certainly do, and spend a lot of time feeling that this is my data and nobody else can look at it until I publish it. That had to go away. That's not to say that you shouldn't acknowledge the contribution of the people who generated data. And that really is the paradigm from Genomic. The Human Genome Project, the Cancer Genome Project, all took that path. And I think there's way, and there will always the bad apples. I'm not saying no one. There will be someone who take advantage of that situation. But you will know who those people are. But I think over time, my hope is that that will prove to be win-win for everyone, because chances are people who use your data for their hypothesis, they're answering a question you hadn't thought about. Over time, on average, that's going to be the case. So with time, I hope that it will prove that it's much better that now you have 500 samples rather than 50, because you're getting everybody else's sample for your question, because someone else is benefiting from your 50 sample. But it will be a transition. We need to prove that out. Yes?
So where I was going with that is, how are you actually, sort of patterning the chromatin to say, okay, this patient is different than this patient? Are you using some sort of, platform to analyze their epigenomes, which we know obviously is very complex with all the modifications? So I'm just curious if you could speak a little bit to how you're sort of assigning these, epigenomes, and if they correlate at all. This is actually, if they correlate at all with the genetic status, if you know it. And I know you've done a handful of patients. It may be hard to say.
Okay, so first, the tech platform. The validation study that we looked at the Nevi that I show you only looked at 4 genomic regions, 4 regions in the genome, with, I believe, 6 different marks, chromatin marks. And we basically determined the status of those marks in those 4 regions across the sample. And then you can treat them as any other data point, whether it's expression data or copy number data. And what we did was a unsupervised hierarchical clustering to show how they cluster together on one side, and the other one was a PCA analysis to show them. So they are simply a readout of the enrichment of that mark in that genomic regions. And when we do whole genome ChIP-seq, we're looking at enrichment of that mark at that particular genomic region across the whole genome. So obviously there's many more data points. And so what we are now doing is taking the tumor sample and do the full genome sequencing, ChIP-seq, so that we can begin to correlate that to the genomic information. On the sample that we looked at so far, we know, I don't have the slide there, we know the BRAF and NRAS status of those samples. That's about it. And there isn't anything that pop out, but I wouldn't expect, given how small the sample is, among the number of variables we're actually looking at. So right now, I can't say anything about it. There is another analysis we have done that's more sophisticated, and we really are looking not just at chromatin mark, we're defining the chromatin state. And what, I took those slides out, but what we found is that it's not a chromatin mark independently that dictate the regulation of gene at that moment. It's actually a chromatin state, which is a permutation, a combination of multiple marks in that regions across the 36 we looked at. Now you become really complex, and we're looking at, how many states you're talking about, but we clearly can define, activated state to, quiescent states based on the chromatin structure, and it matches with the genomic annotation that we know about. And so that itself represents a way to define and discover a new element in the genome. So what we're hoping to do with that data is, I think what we like to do is get that data published so that it become a resource for the community, because we only did that 1 analysis or 2 analysis. The truth is there's probably a whole lot more that can be done with the data analysis, particularly when we start having them on the tumor samples.